clones from southern Ecuador have shown relatively poor results with IGF. But I see this happening because my strong belief is, as is shared by some of the pediatric endocrinologists the world over, that some component of so-called idiopathic short stature may be actually a milder form of growth hormone insensitivity. I see this area emerging in the years to come. Next slide. Now what do I visualize for the future? Future has a lot to offer and a lot that I cannot visualize now. But what do I visualize? I visualize a lot of shift to preventive endocrinology as likely to happen everywhere, and totally new and weird vistas of understanding of endocrinology are going to come up, primarily with the coming up of the Human Genome Project, where better understanding of the genetics of many of the endocrinopathies are likely to be better understood, and corrective measures are likely to be put in place so that some of these devastating things do not perpetuate. Next slide. And last but not the least, we will continue to be in the middle of understanding things. Last chapter will never be written. That will be the dead end of the whole thing. And I visualize areas really coming up dependent on, in endocrinology, I emerge areas developing depending on what is happening in genetics and what is happening in immunology and basic sciences. Whatever happens there, which as a pure clinician I cannot imagine, is going to have a direct bearing how the practice of clinical endocrinology is going to change in years to come. Thank you very much. Any questions? I hope I stuck to 20 minutes.
Yes, thank you. There are no questions or comments.
One question, but it needs maturation division. It takes three months probably to pass the ejaculatory tubes and epididymis. Will there be any fault with the, this ovum and the sperm conjugation? I want to clear this.
You see, basically, I first said this is what I speculate. This is what I speculate. Now, what is happening is that if you are able to get a cell that has already undergone a meiosis and has the potential of ultimately turning into a good supermetazoa, if because of any reason this is not able to be made available to an oocyte, I visualize this directly getting implanted into the oocyte. Yes, people have raised a lot of queries, and one of the major queries that has been raised is that how do you understand that this spermatate is not sick and you will not have a higher prevalence of congenital anomalies in them? So that's what I said. By the time the Human Genome Project gets complete, and you know you are sort of sanitizing and sterilizing the sperm, then only allowing it to go ahead.
After going through the literature and going through your lecture, there is no hope that we can prevent to get diabetes. Can you please elaborate this?
You see, treatment. When I treat a patient, or when any one of us, very senior people, will treat a patient, we never say that we are giving him an eternity. We are prolonging his life. We are worsening, or we are improving his misery. What I visualize is that 80% of this diabetes is directly linked to two things. 80%, not all, minuscule cases here and there. The genetics and obesity. You take care of obesity, you may not, I underlined, you may not say no to diabetes. But if a person has a weight of 100 kgs and his height is, let's say, 170 centimeters, if by his genetics, his destiny to develop diabetes, it will happen at the age of 30. Reduce his weight to 80 kgs, it will happen at the age of 40. Reduce his weight to 70 kgs, it will happen at the age of 45. Reduce his weight to what is optimal for him, let's say 66, it will happen at the age of 70. So this is what I meant. You have already doubled his time of non-diabetic period. One. Second thing, in that group of subjects where you understand genetics. Third, for the clinicians, when you diagnose a person to have diabetes and you look at what is happening to his beta cells, to his insulin receptors, and look at it retrospectively, studies have unequivocally shown that the process that ultimately leads to impaired glucose tolerance or diabetes has been in place for a decade. It means nature had already given a decade for intervening. I visualize this getting diagnosed during that period so that this decade becomes couple of decades, 4 decades, 3 decades. But I never mean that you will have zero diabetes on this planet. We're not talking about these 300 million people. We are only talking of trying to reduce it to 100, 150. We're not talking of making it a non-entity.
Diabetes also sign symptoms, please clarify.
That was what I was trying to tell you is what I visualize would be happening in relation to delivery of growth hormone to human beings. That patients with Fanconi syndrome are, I visualize, going to be tried with growth hormone, not with diabetes. Because there is a belief that there is a higher prevalence of innate biological growth hormone deficiency in patients with Fanconi syndrome. That innate needs to be discussed. It takes some bit of time. For those additional 5 minutes, thanks a lot.
Talk on recent trends in HIV infection. Professor Mustafa Siddiqui did his bachelor's and master's degrees in science from University of Kashmir in 1973 and 1975 respectively. He carried out research leading to He did his postdoctoral fellowship in molecular immunology under the prestigious Monbosho Society of Japan in Shiba University, Japan in 1983. He was awarded Alexander von Humboldt, I hope I'm pronouncing it correctly, fellowship in Germany in 1991 to carry out research for 2 years at Department of Immunology, University of Ulm, Germany. He was a visiting professor at Paul Ehrlich Institute, Frankfurt, in Germany for 1 year in 1998. He has more than 25 international publications to his credit, which include the one recently published in the prestigious Journal of Biological Chemistry, and this happens to be his favorite publication till date, Technology, Government of India. He is also the recipient of an equipment grant from Germany to study single cell functions on FAC scan. Professor Mustafa Siddiqui presently holds the chair of Professor and Head Department of Clinical Biochemistry at Sher Kashmir Institute of Medical Sciences. I think for the time being, this should be enough. May I now invite Professor Siddiqui to deliver his lecture.
Chairpersons, I would be brings Millennium to the HIV. I'm too small for that. But I would say what has happened during the last 1.5 decade, during which time we have experienced the HIV scourge, and what has happened recently, because there is too much of uncertainty in the field of HIV that can be predicted by clinicians or basic scientists together. I would just give a brief introduction. What has happened to the human species? Human species has existed something like in the present form for about 200,000 years. And during that time, the immune system has presumably played a critical role in our ability to weather exposure to parasites, bacteria, viruses, toxins, and other hazardous. The time that has decided about the relationship between these external agents, whether they come in the bacterial form or viral form or whatever form, and how human body has behaved, immune system has behaved. Ultimately, it has resulted in a kind of existence of both the species, both the microbe as well as the human beings. But this is the optimism that we should live with about the HIV scourge. That means for 200,000 years, if human body has lived all through these years, then obviously we may probably even sustain the HIV infection. But it's only an optimism. But the way that it's taking the death toll of the people who are infected by HIV, we may still find that it will be one of the major death causes in whole of the world, especially in the developing world. Immune system has no longer enjoyed the luxury of time. Air travel major population growth and emerging megacities, they have vastly increased the ease with which people have become exposed to agents of disease. Let me go to the first slide, please. Right here, and you can see almost whole of the world was diseased by the H3N2 influenza virus by only somewhere in 1991. So this is actually the air travel, and obviously, as we call the global village, the whole globe has become, and we have been exposed to more of the diseases, and the scourge will be more in focus about HIV. And if you look at the HIV also, HIV infection per person, and it started from the California, that is the west coast of the United States, and almost it was reported in 1981 in the Californian homosexuals, and it is now this time affecting almost whole of the globe, and no part of the globe can claim immunity against the HIV virus or HIV infection at the moment. That means within 12 years, whole of the world reported HIV. That means within 12 years from the United States, which started the reporting of the first AIDS cases, whole of the world is infected. Next slide, please. Of course, since the transmission causes, we know the route of the transmission, so therefore it actually works out to be in the age group of 10 to 24, because it is the most sexually active group or the age. Therefore, 20% of the infections are projected to be occurring only in this age group. Next, please. This is the global scenario of the 1994, and since then we have a lot of changes. We go to the next slide, please. This is how in the Africa or even in the West, in the Asian countries, the heterosexual transmission is the major cause of spread of the HIV. Next, please. Next, please. Okay, I go to the basic biology because I'm just trying to talk about the gene therapy and the immunoprophylaxis of the HIV. Unless and until we understand some of the constituents of this HIV virus, we may not be able to understand ultimately gene therapy. I took up this task of explaining a little bit of gene therapy because it's often being talked about in medical journals. Therefore, I thought I'll just touch it on the brims. So this is the main structure of the HIV, which is just a diagrammatic structure. We have on the coat or the envelope protein, we have the GP120. It's mostly talked about. 100 GP and GP120 is an envelope protein with which this virus gains entry into its host cell that we come to later. And it's an RNA virus. That means whole genetic information is carried in RNA rather than the DNA, which is the normal. DNA is the normal genetic information site of any organism. Of course, we have the RNAs, can also be done, can also have the genetic information. So this is one of the unfortunate for human beings, that it happens to be attacking the very basic cell that works out for the immune system, and at the same time it's an RNA virus. If it was a DNA virus and it would still attack an immune-mediated cell, we would have by now the cure for the HIV. So it's an RNA virus, and whole of the RNA that is here is being transcribed into the DNA by a system. Next slide, please. So this is the RNA which is present in this whole virus. We have what we call the pole here, and we have the reverse transcriptase. Reverse transcriptase is an enzyme which can convert the RNA into the DNA. But unfortunately, all the enzyme systems that are present in the human body have a very proofreading system. We are human beings, and our children are also human beings without any major mutation. The reason is that whatever enzymes are working during the replication, that means the DNA formation of another DNA copy, there is a proofreading system. We call it fidelity. That means we preserve the DNA structure of the human beings. That's how we are in the same form for the last 200,000 years. But what happens is, in case of the reverse transcriptase, that is the reverse transcriptase here, it's not a very good enzyme. When it makes a DNA copy out of the RNA copy here, the reverse transcriptase makes errors. And when it makes errors, it is, is passed on to the next generation. That is how we find a real difficulty in making a drug against this HIV virus, in making a vaccine against this HIV virus. And also, a very phenomenon that means, for example, a person is infected 3 years earlier, and if you look at the HIV structure now, after 3 years, you will find it's a totally different one when it entered the body. The reason is, when the RNA, when the virus makes another copy of the virus within the T cell, within the CD4 positive cell, when it goes back to make the DNA copy, it makes errors. So you just introduce the mutations. That's how we call the RNA viruses are highly mutagenic. Therefore, the making of a vaccine against this virus is extremely difficult, and that's why we do not have a very successful virus even at this moment. Next, please. So the three routes of HIV transmission, they include the sexual and the parenteral, as well as the perinatal. Sexual, we know that it started with homosexuals, although this time too, I mean in the developed world, especially from the West Europe and the North America, it is the major cause of the transmission, that is the homosexual. But in the developing countries, especially in the Asian countries, it is mostly the heterosexual that constitutes the major transmission mode. And then of course the blood transfusion, needle sharing, among the IDU, needle stick injuries and exposure to open wounds or mucous membranes, and perinatal is intrauterine and peripartum. Next, please. This I just want to touch on the periphery again is that what is why does this about the CD4 is it's a helper T cell of the immune function. The helper T cell function has a major function of not only promoting the antibody production by the B cells or the plasma cells, but also helps the other forms of the T cells, like CD8 cells that are cytotoxic in nature against the viral infections. But unfortunately, this CD4 T cell, it became the target for the HIV virus. And normally, this CD4 acts in this fashion in the immune system. Next, please. If you look at the life cycle of this HIV virus, it's simply this is a CD4 positive cell or the helper T cell. When it attacks itself to the CD4 antigen that is displaced on this T cell, therefore naming it as the CD4 positive cell, HIV virus is internalized by fusion, and thereafter it makes a single-stranded RNA copy, after which, again I said that the reverse transcription, which the reverse transcriptase enzyme, which is present already in the virus, it makes a double DNA copy, double-stranded DNA copy, and ultimately it's being transported to the nucleus, through the nuclear membrane, where it gets integrated into the human genome or the DNA molecules of the normal host cell. And ultimately, when it stays here. That's what we call the clinical latency. From this point up to this point, it takes only 2 to 3 days from the infection to the integration into the human gene or the human genome. Then it stays there. Then what are the factors to break this clinical latency? And clinical latency can vary from 6 months to even now we know there are people who are living with AIDS for the last 18 years. I'm going to touch the long-term survivors of the HIV. And there are then certain factors which are T cell activators which somehow break this clinical latency and ultimately makes it to express again. So it makes further copies of RNAs when our virion assembly is there. Ultimately there is budding and you have further virion particles in the plasma ready for infection for the another T cell. Next please. Okay, this is the chart that shows the clinical latency, the primary infection period, clinical latency, and then the disease. If you look at this graph of the CD4 positive cells, we have the CD4 positive cells more than 1,000 per cubic millimeter in a normal human body, or even we can say per microliter. When infection is there, HIV infection is there, we call it seroconversion. The CD4 cells is being taken up or rather destroyed by the infection of the HIV molecule, and ultimately it rises again because there is already a mechanism called as hematopoiesis which tries to retain the number of the CD4 cells, and ultimately it rises again. Then you have an asymptomatic period which is called as the clinical latency, ranging up to 12 years, but even now 18 years is also possible. And then you get the symptoms of the purely of the HIV, starts getting symptoms, infections, and then the final AIDS. And if you look at during this period, what we can find out is the antibody titer to this HIV is going up, and then ultimately it reaches the plateau when it starts decreasing, but never decreases. Even at the time of the death of such patients, you have the HIV antibodies against. Next, please. This is which normally takes care of the viral infections. So the loss of this is due to the direct HIV-mediated cytopathic effect. When there is a budding of viral molecules from the CD4 cells, it makes holes in the plasma membrane. Ultimately, there is the death of that CD cell. Second is syncytia formation or syncytia formation and cell death. What happens is that because of the, when the DNA of this virus, it gets integrated into the human genome, it sometimes starts expressing it on the cell surface. So some of the CD4 cells are expressing the GP120 molecule of the virus. So because of this, there's an interaction between this cell and a new fresh CD4 positive cell. Ultimately, you have large multinucleated cells which are forming the syncytia, and ultimately it leads to their death. And then we have an apoptosis, that's a programmed cell death because of the formation of very new protein molecules on the cell surface. So there's a programmed cell death. The immune system doesn't want to see it growing, so therefore it tends to make it die. Next, please. These are the clinical stages of the HIV disease, in which we have early stage 1 is just a primary infection, we cannot detect it, and the stage 2 is when the CD4 count goes still more than 500 per cubic millimeter. Stage 3 is when it goes to 200, it's not 2000, I'm sorry, it's 200 to 500 per cubic millimeter, and late stages hardly there are 50 to 200 cells, and advanced stages when they start dying is less than 50 per cubic millimeter. Next, please. Now, who are the long-term survivors, and what's there something very strong in these long-term survivors which are harboring the infection but are not dying or not getting any opportunistic infection? Recently, there has been found to be, the CD4 has been found to be as a receptor for the HIV, but there has been found a co-receptor. As a chemokine receptor, we call it CCR5 and CXCR4, which are normally present for other immune functions, but this virus also works or enters the cell through these co-receptors. Next, please. Infect you to HIV, I have an answer for that. And next, please. Gene therapy models are either prophylactic or therapeutic. Next, please. This is the carrier for the gene, in which we introduce the gene, and ultimately when we introduce the gene, we push it into a very appropriate cell so that it expresses in preference to the older gene which was existing there. Next please. The prophylactic genes are mostly based for HIV on the ribozyme, which degrades the RNA of the HIV virus. Therefore, we introduce this ribozyme gene into the hematopoietic cells or into the cells, which ultimately lead to the formation of the CD4 positive cells. Therefore, when the HIV virus comes in and it starts transcription to the DNA, this ribozyme will completely clear the RNA of the virus, therefore will not allow its infection to proceed. Next please. The therapeutic is my own model which I have proposed and I have worked on it. I have already published several papers on this. That is, I have brought in, based on the structural changes in the receptor and co-receptor of the HIV, I have brought in structural change in the CD4, and ultimately at the moment I have a gene which is non-infective to HIV virus and is having a normal function. Next please. This was the schematic diagram for my own gene therapy model. That means if we are somehow introducing a gene which is not infective to the HIV virus, if we introduce it into the T cell precursors, when they go into the thymocytes, we will have mature CD4 cells which will express mutant CD4, not the original CD4 which is infective to HIV, but the mutant CD4. Next please. And this is some of the experiments showing that this mutant number 3 is not infective. That means it's not expressing the virus at all. P24 is an indicator of the expression of the virus in the cell. This M3 is not infective to HIV virus. Next please. So immunoprophylaxis, that means about the vaccines. They are mostly based on whatever is going on in the clinical trial 1 and 2. They are mostly based on the envelope GB120 protein. Clinical trials at the moment are more confusing than otherwise. The reason is that in the same group of patients, some people, because most of the patients that were selected for this were already having the HIV infection. So there is nothing like that. We would first prime the body against with this vaccine and ultimately bring in the infection, because that's not possible in HIV. So the volunteers were selected on the basis of that they already had an infection of HIV, and then this GP120 based vaccines were given to them. But the data suggests that they are more confusing than they can find that exactly that they will come to the market. Next please. So ultimately at the end of the 2001, we still have to go to the preventive measures for control. That is the educational approach, behavioral approach, technical approach, and regulatory approach. Educational approach, I would say that we have to educate the people about that still there is no cure for HIV. So they have to change their behavior. They have to change their behavior. And the technical approach is what the professionals can do, how we can stop the cross-infection of HIV. And the regulatory approach goes to the government's head, how they regulate the HIV infection. Thank you very much.
Are there any questions or any comments? Dr. Wahid.
In that the virus goes to CD4. CD4 and not to the receptor, co-receptor, CCR5. Can we activate the receptor activity or block the entry of virus to CD4? Is there an area like that?
Yes, in fact, if you see the zidovudine mode of action of the zidovudine, that is AZT, the mode of action is simply because these are dideoxythymidine. It's a dideoxythymidine which terminates the DNA synthesis. So when from RNA you start making the copy of a DNA by the use of the reverse transcriptase. So when it starts that, you introduce the AZT so that there is no DNA copies, because it stops the elongation of the chain. That is the one. But why we are not able to stop the entry of the HIV, if you look at one of the clinical trials that went was to inject into the patients the soluble CD4, so that there is a competition between the CD4 cell and the soluble CD4 with the GP120. But it brought in major problems of immune complex related problems. So it became a very big molecule. Virus is a very big molecule, and CD4 also is a very big molecule. So it became too big to pass through the capillaries and all that. So you had immune complex related problems like illness, which activated the inflammatory process. So it was abandoned because of that. It's not long. Actually, in some people, you will see that this clinical latency, it changes only from 6 months to, as I said, that still the people are living from the first report. The clinical breaking of the clinical latency is dependent on several T cell activation factors. Like, for example, other viral infections. If a patient at this moment of time is having a lot of herpes infections, herpes simplex virus 6 infections, you will see that his clinical latency is broken much faster than the other one who has not the herpes simplex virus infection. Because ultimately it depends upon the activation state of the T cells. That will decide upon the clinical latency period.
This mutant CD4 cell, whatever the normal function, you
Normal?
will preserve the normal function in mutant CD4.
In my one,
Yeah.
yes, we have done that. There is a lot of technical, I didn't touch it here. We have preserved basically, the CD4 has a function of the class 2 MHC binding to the macrophage and all that. We have preserved that. I mean, we have at least mutant of about 35, out of which only one mutant is, which has preserved the original CD4 function, normally which is, and is non-infective to HIV.
So why would it be easier to tackle the HIV virus if it would have been a DNA virus rather than the HIV virus, which you just mentioned?
Because, as I said, that there are so many DNA polymerases which preserve the fidelity of the replication. But in case of reverse transcriptase, it's not there. So it's somehow there is a mismatch, base which enters, and ultimately there is no proofreading by the reverse transcriptase. That's why we have a problem at this time of point. If there is somebody is infected now, and after 4 years or 5 years from the same patient, you will find a different phenotype of the HIV virus. It's only because this reverse transcriptase introduces the mismatch pairs, and there is no proofreading system.
One more question, Dr. Mustafa. As you said, we know that all the retroviruses, including HIV virus, possesses the reverse transcriptase. What are the present trends to stop this reverse transcriptase enzyme?
I said the ribozyme, one of the things is gene therapy model based on the ribozymes, because ribozyme is a catalytic RNA enzyme which will break these RNAs only. So they want to, she's a very Flossie Wong's fellow, her group is working very hard on this, is that if we can activate, when the cell gets infected by HIV and it starts reverse transcribing the RNA into the DNA, this ribozyme should work and it should cleave the whole of the RNA so that there is no formation of the DNA copy from this and ultimately integration into the human genome.
Dr. Khurshid Iqbal, he'll talk on the coronary epidemic. Dr. Khurshid Iqbal graduated in medicine from Government Medical College, Srinagar. Graduated in 1977 from Government Medical College, Srinagar for obtaining the maximum aggregate marks from first to final MBBS. He obtained his DM cardiology from the prestigious PGI Chandigarh in the year 1988. He has approximately 28 international and national research publications to his credit. Dr. Khurshid Iqbal has designed and fabricated an indigenous external pacemaker which has been tried and found useful clinically. He is proficient in non-invasive and invasive cardiology and started dual chamber pacemaker implantation at SKIMS. Dr. Khurshid Iqbal, may I take the liberty to call you our IT man, for he gets the credit of establishing the Medline services and the SKIMS website. Ladies and gentlemen, may I now invite Dr. Khurshid Iqbal to deliver his lecture on the coronary epidemic.
Good afternoon, ladies and gentlemen. Thank you very much, Dr. Khan, to give. Actually, you also made me feel like Professor Zargar embarrassed with this elaborate introduction. I chose this topic, the coronary epidemic, because I feel there is lack of understanding of the magnitude of this problem and about the risk factors and how they interplay. and we have this epidemic and it's actually real, and it's come to J&K also. It's very important to know the magnitude, because then we'll know the exact figures and exact numbers that are going to affect us in the decades to come. Secondly, we also should understand how these risk factors interplay and how we can pinpoint a particular individual or a group of individuals who may not have the disease, but we can say, yes, this individual or this group of individuals is likely to get affected by coronary artery disease. And most importantly, the treatment of established coronary artery disease is out of reach of most of the developing countries. We cannot afford it. We don't have infrastructure and it's very expensive. Most of our patients will not be able to afford it. So the thrust has to be on prevention. As Professor Zargar also said, the next millennium, next 10 to 20 years, there will be more and more spending on preventive aspects of most of the diseases, and coronary artery disease is possibly the number one. May I have the first slide, please? The CVD burden. I want to concentrate the audience only here. If you compare the developed and the developing nations, here we have India and China. The population of India, these figures are of 1990, and we know the population of India has already crossed 1 billion. So let's take this 90 as an example, and India population 849 million and China 1133 million. Here you see the coronary artery disease mortality is more than China, although the cardiovascular disease mortality, which includes peripheral vascular disease, strokes, is much more in China, and that's why the figures slightly higher than India. This figure is changing in China. It's approaching India. Meaning that the coronary artery disease is increasing disproportionately compared to other cardiovascular diseases like strokes even in China. Then I also want to concentrate you on in this last area, which is the percentage of the total cardiovascular disease death throughout the world. If you combine established economies and former socialist economies, this comes to about 37%. That means 2/3 is contributed by the developed world, even now. And this scene is going to change. In next 10 to 20 years, we are going to go through a full-scale and a mature epidemic in developing countries. May I have the next slide, please? So why we are getting this epidemic? We have been saying that our diet has changed, lifestyle has changed. We are quite vague about it, but we have scientific evidence and hypothesis to say why this change is coming here also. The epidemic is already in United States and developed countries where it's mature, and the incidence of coronary artery disease is falling there. It had crossed 50%, now it is falling. Here it's much lower, but it's going up. This is a model of epidemiologic transition, which actually explains most of the demographic changes related to most of the diseases. Here we are referring to coronary artery disease. It is that the countries or the populations pass through phases. The phase 1 is called age of pestilence and famine, underdeveloped places, where cardiovascular deaths are only 5 to 10%. And of those also, the majority belong to nutritional diseases, infectious diseases. Asthmatic heart disease is an important cause of cardiovascular death. And the countries at this time that are passing through this phase are Sub-Saharan Africa, some parts of South Asia, and South America. Then we have the next phase, age of receding pandemic, that we are controlling the nutritional and infectious diseases. Affluence is coming, standard of living is going up, and processed foods are consumed more, lifestyle is becoming sedentary, the cardiovascular diseases, hypertension and related diseases start coming up. And overall cardiovascular disease burden increases to 10 to 35%. Africa, North Asia and South America belong to this phase, right now. Where are we figuring? We are figuring in phase 3, degenerative and man-made disease phase, where the coronary artery disease or cardiovascular disease in total account for 35 to 65% of death, and they are contributed by coronary heart disease, stroke, peripheral vascular disease. South America, Asia, and Eastern Europe belong to this. A word about Eastern Europe: there we have much more homogenized involvement. In South America, Asia, and Asia, what is seen is that higher strata of the society is affected more, while the lower strata is affected less. But they are coming up. More and more affluence, more and more economic development and industrialization makes the societies more homogenized, as has happened in Eastern Europe. Then we have phase 4, called phase of delayed degenerative disease, where the coronary artery disease, cardiovascular disease causing death starts coming down, where it is less than 50%. We have decreasing incidence of cardiovascular disease, and this is because of multiple factors. Most important is widespread and proper medical education, health delivery systems, and proper management of coronary artery disease. And North America and Western Europe and New Zealand are the examples. We have the next slide, please. So when we come to the subcontinent, so what are the various causes or various reasons why this epidemic has actually come up? Continuing with the demographic changes, what I said, we have a population expansion because the life expectancy has gone up. So more and more people that are likely to get coronary artery disease, their absolute numbers increase. So we have an increased absolute numbers of the population that are susceptible to coronary artery disease. So it adds to the pool. An estimated 300 million in developing countries out of 500 million total are susceptible to coronary artery disease. And it has been estimated by year 2020, the population of adults more than 35 will increase from 28% to 42%. And we know in South Asia, coronary artery disease comes quite earlier. So you have a very large pool, at 42% of population which is susceptible to coronary artery disease. The increase in living standards, advanced lifestyle, increased smoking, blood pressure, cholesterol, and sedentary habits are further contributing to this epidemic. Next slide, please. Another thing which has been postulated to cause an increasing number of coronary artery disease patients in South Asia is 50 gene. What is it? It is said that there are adverse environment of limited nutritional resources leads to genetic programming for survival over generations. So when there is a change in the society, this particular individual becomes susceptible. There is a mismatch. What happens? A gene which actually increases the efficiency, say, of fat storage through an oversecretion of insulin will now cause obesity, hyperinsulinemia, diabetes, and atherosclerosis. And something which is related to it is another hypothesis called fetal origins hypothesis, where here it's occurring in generations, here it may occur in the same generation, and adverse intrauterine influence due to poor maternal nutrition may lead to impaired growth and low birth weight. And when the same individual, when he is exposed to a better environment in later life, the adaptive metabolic and physiological response to facilitate survival become actually a stigma for him. He cannot cope up the challenges, say he may be exposed to higher salt, higher fat intake, and so on, and he may become susceptible to an increasing incidence of hypertension, coronary artery disease, diabetes, and so on. Next slide, please. Then a word about ethnic diversity. What we have seen that South Asians that migrated to UK had much higher incidence of coronary artery disease. Same was true about Japanese who migrated to US. But there was a difference. If the environment is common, but gene pool is different, say South Asians going into England. It is the non-conventional risk factors that become important because they are exposed to the same conventional risk factors because their diet is practically similar, the environment is practically similar, the stresses are nearly similar. But if gene pool is common but environment is different, then the conventional risk factors become more important. Next slide please. So we come to conventional risk factors: advancing age, male sex and postmenopausal females, hypertension, diabetes and hypercholesterolemia, smoking, obesity and sedentary living, and positive family history. I am not going into the details. Next slide please. The non-conventional risk factors which have definitely an influence in increasing the incidence and prevalence of coronary artery disease in South Asia are a triad of hypertriglyceridemia, low LDL cholesterol and high LDL cholesterol. And high LDL cholesterol, I don't mean an alarmingly high, mildly elevated LDL cholesterol, but a low HDL cholesterol and an adverse ratio. They are prone to coronary artery disease, even though their total cholesterol and low density lipocholesterol by conventional definitions may be normal. Then a higher prevalence of lipoprotein small a, insulin resistant state, a different phenotype of LDL, an increased fibrinogen in the blood, and homocysteine, and lastly infections and inflammatory factors. A lot of work is going here, particularly chlamydia, whether it's the infection by chlamydia which initiates the coronary atherosclerosis, or a coronary atherosclerosis becomes worse when they become infected by this chlamydia, is also under investigation. They may be all contributing some percentage, some degree of increase in the incidence may be interplayed by one or more of these factors in addition to the conventional risk factors. Next slide please. A risk factor will create a situation where an apparently normal value may be still responsible for a significant number of individuals who will get disease in future. So actually risk factors operate in a continuum of progression, and lower segment contributes maximum numbers and the lower segments may be actually missed. The reasons for this are multiple: epidemiologic transition, population expansion, changing lifestyle and genetic factors are operating in various proportions. The infrastructure to deal with enormous number of established states is grossly insufficient, and the cost of treating the disease on modern lines is prohibitive. Next slide please. So non-conventional risk factors also play an important role in patients. The threshold for intervention risk factors depends upon whether we are dealing with clinical or population situations. Risk factors work in continuum, and multiple risk factors predict magnified risk. An approach should be aimed at controlling the risk factors in established cases as well as in general population for comprehensive curtailment of coronary disease.
Is there any question, Dr. Wade?
I will try this common risk factors. We know hyperlipidemia is a common risk factor, and what is the role of a doctor in aggravating the disease of the patient? You see, atorvastatin is a very good drug as an anti-lipidemic, hyperlipidemia. But at the same time, it raises fibrinogen levels, number one. Number two, in hypertensive patients, we right and left prescribe beta blockers, and those raise lipids again. So what is the role of a doctor? Number two, what is the role of endocrinologist?
Role of doctor is to give proper education and treatment to the patient. As far as these two drugs are concerned, like you raised the question of atorvastatin. Yes, there was one study which showed an increase of fibrinogen after long treatment with atorvastatin. But there was a much larger study which came later, which has denied that. So as of now, this controversy is not settled. Possibly, if at all it's increasing the fibrinogen level, it's not statistically significant. As far as beta blockers are concerned, yes, they do increase lipids. Here the recommendation is clear. If we are dealing with a hyperlipidemic patient, we better avoid it. But if the lipid levels are normal by the standards which I have given, it should be given. Beta blockers and diuretics for hypertension are the two drugs that have been established to reduce the morbidity and mortality with this. In addition to that, ACE inhibitors are also have started coming in the same group. And what was your next question? Antioxidants, again, the area is great. There is one study which has shown deleterious effect with beta carotene, and we had one study, large randomized.
Thank you very much. There are no questions. I think that was the last question from Dr. Walid, and I have been directed by the director to close it because there is some meeting at 2:30. So thank you very much to all the speakers, Dr. Hamid, Professor Khurshid, and Dr. Mushtaq Siddiqui. Thank you very much.
With this, we come to the end of scientific session. I, on behalf of the organizing committee, would like to thank Professor Qadiri and Professor Wani for chairing the session. May I invite Dr. Mustafa to conduct the valedictory function. Thank you.
Merajudin, Director, Kashmir Institute of Medical Sciences, will preside over the valedictory function. Honorable Director, Dean Medical Faculty, Kashmir Institute of Medical Sciences, ladies and gentlemen, we now come to the valedictory function of this seminar. During the scientific deliberations, we have had the opportunity to listen to some of the experts talking on various important healthcare issues. At the outset, Professor Imtiaz Ali, Head of Department Community Medicine, highlighted the importance of WHO theme this year. Indeed, mental health is fast emerging as a major health care issue. Dr. Amin Tabish spoke at length on various aspects of healthcare planning and management. He focused on some of the serious problems confronting the global population at large and this country in particular. He has rightly emphasized the need to reach out to the deprived and inaccessible segments of population. I do hope that, as suggested, the policymakers at national and state level would meet the WHO recommended guidelines and would earmark at least 5% of the GDP towards healthcare in future. Professor Jalal, Head of Department of Endocrinology, highlighted some of the most important emerging health problems like diabetes mellitus, iodine deficiency disorder, and infertility. He has emphasized the need to address to these problems which exist significantly in Kashmir Valley also through health education, supplementation of the diet, and application of genomics to understand the biology and behavior of these healthcare problems. HIV and AIDS is emerging as one of the major healthcare issues in the developing and underdeveloped world. Professor Siddiqui, at the Department of Clinical Biochemistry, highlighted some of the major social and environmental factors responsible for this dreadful disease. We in this part of the world have recently witnessed an escalation in HIV-related cases. However, with the increasing population movement, we must not leave any stone unturned to educate our population into various preventive aspects of this disorder. Dr. Khurshid Iqbal, Additional Professor of Cardiology, emphasized on early risk factor assessment and coronary artery disease prevention. A country like ours with limited resources, the cost for treating coronary artery disease is astronomical, and we agree with him that it is worthwhile evolving a preventive strategy among the high-risk population. Ladies and gentlemen, we do sincerely hope that deliberations in the scientific session were informative and would help us in evolving a comprehensive healthcare strategy in near future. May I now request Honorable Director, Syed Kashmir Institute of Medical Sciences, to felicitate various participants in this seminar. I request Dr. Musharraf for help, please. It's a pleasure to request Professor Nazir Evani, Head of Department Surgery, to accept a token of appreciation
for his contribution as chairperson in this seminar. Professor Mustafa. It gives me great pleasure to request Professor G.J. Qadiri, Head Department Hospital Administration, to accept a token of appreciation from organizing committee. Professor G.J. Qadiri. May I now request Professor Abdul Hamid Zargar, Head of Department Endocrinology, to accept a small memento. Professor Abdul Hamid Zargar. I now request Professor Mustafa Siddiqui, Head of Department Clinical Biochemistry, to come over and accept a token of appreciation from organizing committee. Professor Mustafa Siddiqui. I, on behalf of organizing committee, would like to request Dr. Khurshid Iqbal, Additional Professor of Cardiology, to accept this memento. Dr. Khurshid, please. Dr. Syed Amin Tabish, would you be kind enough to accept a memento from the organizers? Dr. Amin Tabish. May I now request Program Coordinator Dr. Abdul Salam to accept certificate of appreciation. I would request the organizing committee to accept certificates of appreciation and would request Dr. Nazia to accept the same on behalf of organizing committee. Dr. Nazia, please. Thank you. May I now request Honorable Director to accept a memento from the organizing committee, and I would request Professor G.J. Qadiri to present the same to Director. May I request Honorable Director to speak a few words before concluding this meeting?
I can only say thank you very much and congratulations and best of luck. Thank you.